Hey, my Wholemood students. So today we are having our third lectural tutorial video whereby we are going to be talking about the cell theory, which is a very important topic in biological science. So before I get into this video, I would like you to please hit that like, hit that subscribe, hit that share, and please give me a comment on what you think about the video. With that said, we are going to get into today's video, and I would also like to state that I am a first year student at the University of the Western Cape. So yeah, let's do this. Let's go. A whole entire mood. So the vlogation. So the cell theory. The cell theory emerges. So now the cell theory had components that were first established during the 1700s, 1600s, and then with passage of time, other new components were added into the cell theory. And then the first major components of the cell theory was that each organism is composed of one or more cells, and the cells are the basic units of life, and cells come from pre-existing cells. That was the first components of the cell theory, and then with time, the second and additional information was added into the cell theory, and the information that was added into the cell theory was all cells have the same basic structure. All cells use energy, and all cells contain DNA, and that is the cell theory that emerges. And then now cells are very, very small in a way that we cannot see them with the naked eye. Now we use objects called microscopes, which are objects that magnifies other objects, right? Well, they magnify the specimen that is being studied. So a microscope is an object that magnifies the specimen that is being studied. So we use microscopes to see through animal cells and plant cells, bacterial cells, and all other cells that we have, because we cannot see cells with the naked eye. And then we have different kinds of microscopes, whereby we have light microscopes, which use lights to magnify the specimen, and electrons, which uses electrons to see the specimen being studied. And then from there we have three different types of domains, and then domains are the broadest and most inclusive categories of naming organisms. We have bacteria, archaea, and eukarya. Bacteria and archaea, they are very, very similar to each other in a sense that they are prokaryotic, meaning that they have no membrane-bound organelles and they lack a nucleus, and they are very, very, very small, much more smaller than eukarya. And then now there are two domains, because now the biochemicals that makes up their flagellum, their cell membrane, and all the other components within their cells are different. That's why they're not in the same domain, and that is why we separate them into their own domains, because their biochemicals are not the same. And then we have eukarya. Eukarya is very complex in the sense that it includes four domains, whereby we have protista, we have animalia, we have fungi, and we have plantae. So now bacteria was the first animals that were emerged on planet Earth, and it is the most diverse one, because there is a whole lot more bacteria than any other domain. In kingdom eukarya, like I said, is composed of four domains, and then it is very diverse. It is composed of multicellular cells, unicellular cells. It is very, 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 very diverse. And then now as you can see here, I have two different kinds of cells. I have a bacterial cell and an and a plant cell. So now, for the bacterial cell, we're going to use the bacterial cell to give like a visual representation of what it looks like. And now it's going to represent both bacteria and archaea, because they are very, very closely related, even though they are different, but like they are almost the same. I drew two structures to make this very, very simple, and we are going to label the structures, define the labels, and give definitions. Well, give definitions and functions. And let me state this before we get into it. One thing that you should know for cells, you should know what the cell looks like, you should know the definition of the cell, you should know the chemical composition of the cell, and you should know the function of the cell. Once you know that, you can answer each and every single question about cells. You can answer multiple choice questions, you can answer short questions, and you can answer essay questions, and you can literally just get and grab all those marks about cells, because cells is about defining chemical structure and knowing the function. And if you know that, you are winning. You are coming for those marks, and you are coming for that distinction that we all should be aiming for. All right. And then now, as you can see, this is a bacterial cell, and you can see that it is rod shaped. And then the rod shaped is determined by the cell wall, right? Which plant animal cells lack. Okay. And the cell wall, the cell wall is the outer coverage that protects the cells, and it prevents the cell from bursting if it absorbs too much water. So now from here we can see that the purple layer is the outermost layer, and then that is what we call our cell wall, right? That is our cell wall, and our cell wall is the outermost membrane that protects the cell, and it prevents the cell from bursting if it absorbs too much water. And then another function of the cell wall, it helps keep the cell together, and it gives the cell a definite shape. Yeah, one thing you should know about cells that have a cell wall is that they have like a defined shape, like a very nice defined shape. And cells that lack a cell wall, they don't have a nice defined shape. Like animal cells, they don't have a nice defined shape, but plant cells, bacterial cells, and other cells that have a cell wall, they have a very nice defined shape. Okay. And then now, after the cell wall, you find the first membrane, which is called the cell membrane. Okay. The cell membrane is the one that is covered in orange, written in orange. So now, what a cell membrane does, it protects the cell, and it separates it from its surroundings, and it protects the inner organelles and the cytosol and the DNA and everything that is inside. But then now remember, bacterial cells have no organelles. But then for those that have organelles, the cell membrane protects everything in the cell and separates it from its surrounding. Okay. And then now, if you can see, they are like tail-like appendages. These tail-like appendages are called flagellum. So flagellum is tail-like appendages that allow the cells to move and to swim and it just allows the movement of the cell, right? And then from there we have these little dots, these little mini dots, and these little mini dots are called ribosomes. To me, ribosomes are literally the second most important after DNA, because ribosomes produce proteins, and proteins are needed by each and every cell for the cell to function. Without DNA, without proteins, the cell cannot function, the cell cannot do its work, and the cell literally dies. And nobody wants to have dead cells, because dead cells means a dead organism, and nobody wants to be a dead organism, right? Good. And then now, from there, when we go inside, we see that there's the thing in black. This thing in black is called I'm not sure if my spelling is correct, but it's called a nucleoid. The nucleoid is where the circular DNA of prokaryotes, right, congregates, right? And the DNA of bacteria is circular, and it is very different from the DNA of eukaryotes. Keep that in mind. The DNA of bacteria is circular. It is circular, right? With that said, we have defined everything that has to do with the bacteria and archaea. These two are honestly almost the same thing, so that is why I put them in the same situation. And then now we move on to eukaryotes, which are very complex. As you can see, we have a very nicely drawn animal's plant cell, and then now we are gonna break down this plant cell. And then the first thing that our cells have, like I said, is a cell membrane, which protects the cell and prevents and like from the surroundings and protect everything inside the cell. Okay, so now eukaryotes. Okay, let's do this. So as you can see, these are like adjacent cells that are found around each and every cell. And then now we have adjacent cells, which we're not going to focus on. And then we have the purple thing is the cell wall. One thing you should remember is that animal cells lack the cell wall, which is why they do not have a defined shape. And then plant cells have a cell wall and they have a defined shape. And then the function of the cell wall is to protect the plant from its surrounding and to give the plant a defined shape. To give not the plant, the cell, and then it also hold the cell together, and it prevents it from bursting if it absorbs too much water, right? And then just next to the cell wall, we get the cell membrane. Now, the cell membrane is very, very special because it is a phospholipid bilayer, right? And then, well, it forms a phospholipid bilayer. The cell membrane is a biological membrane that is selectively permeable, which means that some stuff can pass through it and other stuff require protein to pass through it. So then, the cell membrane, when it is in water, it forms itself into a phospholipid bilayer. And remember, a phospholipid is made up of two fatty acids, right? Well, it has a glycogen that is bonded to two fatty acids and a phosphate group. So now the fatty acids are hydrophobic, which means that they are water repellent. They do not like water, and they really do not like water. They do not form bonds with water, and they just do their most to stay away from water. And then the phosphate group is hydrophilic, which means that it is attracted to water and it forms hydrogen bonds with water. So now when the cell membrane it is in water, the phospholipid arranges itself into a phospholipid bilayer, whereby the in outerness forms the outer that is in contact with water, and then that is the phosphate tail, and the hydrophobic fatty acids rearranges themselves inside the the hydrophilic outer part. So like it's just like a sandwich. So in a sandwich you have your two sandwiches and your cheese and your polony in the middle. So now the two sandwiches are like the phosphate groups. They are outside. The phosphate groups are outside, and then inside you get your fatty acids, which are hydrophobic, right? And then the ones out the hydrophilic is attracted to water, then the hydrophobic is not attracted to water. And then now the nonpolar molecules like oxygen and carbon dioxide as well as lipids can pass through this phospholipid bilayer of the membrane, and then polar molecules cannot pass through this bilayer. So the cell membrane makes groups of proteins that allows the polar molecules to go through the membrane or exits the membrane. Then the membrane also have other proteins. So we have transport proteins which create passageways. So we have transport proteins which create passageways through which polar molecules can exit the cell and enter the cell, right? And then we have enzymes. Enzymes are molecules that speed up a chemical reaction without being used. And then we have receptor proteins, which pretty much serve as name tags, and they just tag everything, okay? And then we have adhesion proteins, which allows cells to stick and bind together. We have receptor proteins, which can trigger a response outside of the cell. So transport proteins, they create passageways. Enzyme, they catalyze and speed up chemical reactions. Receptor proteins, they are name tags. Adhesion proteins help cells bind together, and receptor proteins trigger an action outside of the cell. And then from there we have organelles. Organelles are usually specialized sacs of enzymes. Organelles are usually specialized sacs of enzymes. And then we have the endomembrane system. The endomembrane system is a system that makes proteins and lipids and transport them to where they are needed to be used by the cell the most. Okay, and the endomembrane system, it is made up of organelles. So before I tell you about those organelles, we're gonna come back to this and label the whole entire situation. So now, if you can see properly here, we have this round thing with pores, right? So when you look at it, that is our nucleus. The nucleus is the most important part of the cell. The nucleus houses the DNA, which is literally what the cell needs the most, what our body needs the most. The DNA is the informational molecule of life, and it stores genetic material and transforms it to the next generation, and it helps, it is the reason why we have protein synthesis, which we need which is needed by the cell to carry out the cell's function. Okay, so now in the nucleus we have DNA, and then if you can see there's a dot, like a black dot. That black dot is the nucleolus. Contains subunits of ribosomes, which means that it assembles ribosomes. It doesn't make ribosomes, but then it has subunits that is needed to produce the ribosomes. And then around the nucleus, you can see that there are pores. Né? Those pores, we call them the nuclear pore. They are openings that allow RNA to exit cell for protein synthesis after it has copied the information from DNA. And then the nucleus is surrounded by a double membrane system called the nuclear envelope. The nuclear envelope separates the nucleus from the rest of the cell, and it protects the nucleus, and it makes sure that DNA is at all times in protection, because DNA is literally like the queen of the cell. We cannot have problems with the DNA, right? And then within this nucleus, you can see that there's like a channel of this structure, right? Yeah, so now that structure is called the endoplasmic reticulum. The endoplasmic reticulum. The endoplasmic reticulum is pretty much stack of membranes that originate in the nucleus and spread out across the whole entire cell. And then we have rough endoplasmic reticulum, and smooth endoplasmic reticulum. The rough endoplasmic reticulum is called rough because it has a roughened appearance, and the roughened appearance is made so by the ribosomes that are attached on its surfaces. And then the smooth one does not have a rough surface because it is not attached by ribosomes. And then the function of the rough one is that it holds ribosomes, and then the ribosomes obviously they produce proteins. So now it holds ribosomes and it helps in the production of proteins. And then the smooth one, it synthesizes and makes lipids, and it detoxifies toxins. I'm really sorry about the dance. Then it detoxifies toxins. And then, like I said, the rough one has those ribosomes, and the ribosomes, they make proteins, okay? And then when we go on, you can see this structure. This structure is called the Golgi Apparatus. The Golgi Apparatus. Pretty much the Golgi Apparatus is also a stack of flattened membranes, and then it functions to process and package material, lipids and proteins that are ready for export and that are ready to be used by the cell. Okay, and then around next to the structure, we can see there's many round things which we call the lysosomes. Most plant cells lack the lysosomes, but some plant cells have lysosomes, and the lysosomes, they actually will get to lysosomes. And then also we have the mitochondrion, we have the vacuole, we have the chloroplast, and we have peroxisomes, and then we also have the cytoskeleton, right? And then with the video, we will describe each and every single thing. Now I'm gonna go back to the endomembrane system. The endomembrane system, it is a system whereby proteins are made, and they're exported out of the cell. It starts in the nucleus, whereby DNA, RNA transcribes, gets information from DNA, and then it exits the nucleus. Then when it exits the nucleus, it reaches the endoplasmic reticulum, whereby proteins are made. Then after the proteins are made, it moves from the rough endoplasmic reticulum, where proteins are made, on the ribosomes, to the smooth one, where lipids are synthesized and made. And then after your proteins and your lipids are made, the cargo moves out of the endoplasmic reticulum through vesicles, and then the vesicles transport the cargo to the Golgi Apparatus, whereby the cargo is packaged, and after it is packaged, it moves through the lysosomes, and then it gets to the cell membrane, where it is ready for export out of the cell, and the proteins and the lipids are ready to do their job. Okay, and then that is the endomembrane system. And the endomembrane system, it usually is the question that they ask is the question of milk secretion in mammals. So when they ask you that question, you just explain the mechanism that I just explained now. Then from there we go to digestion centers. All right, digestion centers, we have lysosomes, we have peroxisomes, and we have vacuoles. So we're going to start with lysosomes. Lysosomes are organelles that lyse, right? They cut down. That is why they're called lysosomes. And then lysosomes, one thing you should know is that they contain enzymes that dismantle and recycle food particles, debris, and worn out organelles, right? So now, like I said, they are called lysosomes because they lyse. And then we go on to vacuoles, which and back to the lysosomes. Lysosomes originate in the Golgi Apparatus, but then the proteins that makes the lysosomes are found in the endoplasmic reticulum. But then the lysosomes themselves originate in the Golgi Apparatus. But what makes the proteins that make them are found on the rough Right. Then we go to vacuoles, which are only found in plant cells. Vacuoles are like these structures. Even though I have a small one, vacuoles are usually the biggest organelle in a plant cell, and that is because it produces about 90% of the growth of the cell, and usually it contains a pressure called Tagu pressure, and they also have the same function as the lysosome. Then we also have peroxisomes, which also function the same way as lysosomes, but then now peroxisomes originate in the endoplasmic reticulum and not in the Golgi apparatus, and then they also detoxify toxins, and they remove hydrogen peroxide, which is toxic to the body. And one thing to be aware of about lysosomes is that the lysosomes are very acidic. They usually have a pH of 4.5, which is very different than the pH of the entire cell. If the lysosomes were to burst, the pH of the lysosomes would be different. If the lysosomes were to burst, the reason why they would not cut down the whole entire cell is that the pH of the lysosomes is much more acidic than the pH of the neutral cell. So now when they burst, the pH of the lysosomes are no longer at its optimum, and then now they cannot perform their function, which is the reason why it doesn't cut down everything in the cell when the lysosome burst, or if it was to burst, right? And then we also have the cytoskeleton. The cytoskeleton, it has many functions. It aids in cell division. It helps the cell to maintain its 3D structure, and it does a whole lot of functions in the cell. And then we have three types of cytoskeletons. We have the microfilament. The microfilament is the thinnest one of them all, and it is composed of the protein adhesion. And then the next one is the intermediate filament, which is literally intermediate between the microfilament and the microtubule. Then we have the microtubule, which is the biggest one of them all, and it is composed of the protein tubulin. Okay. And then we also have junctions, and junctions help cells to stick together. And then we also have cell walls, which are found in plant cells, not in animal cells. And like I explained, cell walls help keep the plant together, gives it its shape, and it prevents the plant from bursting if it was to absorb too much water. And then two special mentions are the mitochondria and the chloroplast. So the mitochondria is the organelle that is responsible for producing ATP, and ATP is very, 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 very important because it provides the energy that makes a cell perform its functions. So now a mitochondria is composed of two membranes, an outer membrane and an inner membrane. Then the inner membrane is intricately folded, and it encloses the mitochondrial matrix, right? And then the space between the inner membrane and the outer membrane is called the intermembrane compartment, and then folds of inner membrane are called cristae, right? And then the mitochondria, like I said, it helps with it actually makes cellular respiration, which we need to which is needed by the cell to perform its functions. And then we have the chloroplast. The chloroplast is found in plant cells only, and it is composed of also two membranes, an outer membrane and an inner membrane. Then within the inner membrane we get a structure called the stroma, which is the liquid part of the cytoplasm, of the chloroplast. Then within the stroma we get stacks of grani, and then the grani are stacked into thylakoids, and then the thylakoids are what captures the energy from the sun that the chloroplast uses to produce photosynthesis. Okay, and yeah, those are the chloroplast and the mitochondria are very, very important in the cell. And the chloroplast and the mitochondria have their own ribosomes and have their own DNA, and they can produce their own things. They don't need the nucleus because they have their own things. So yeah, and then one thing I didn't mention for some odd reason is that all plants, literally all plants, have a cytoplasm. A cytoplasm is the fluid inside the cell. And that concludes our video about cell theory. Thank you very much for watching this video. I hope you learned something. I hope it helped you out, and I really, really appreciate it. The next video is going to be about the use of energy, and it is going to be uploaded very, very shortly. Thank you for watching. It really, really means the world to me. Please share the video, please give a like, and please leave a comment, and I'll see you in the next video. Thank you very much.